Hello students, this is Lohita. In today's NEET class, we will be learning about packaging of DNA helix. Okay, we will be learning about packaging of DNA helix. Okay, we will be learning about packaging of DNA helix. So, students, we can calculate the length of the DNA by two factors that we know. Okay, we have already learned that the distance between nucleotides, is 0.34 nanometers, which is equal to 0.34 into 10 to the power of minus 9 meters. 0.34 into 10 to the power of minus 9 meters is the distance between nucleotides, right? And we also know the number of nucleotides. We know that the number of nucleotides in human DNA is 6.6 into 10 to the power of 9 base pairs. Okay, we know that human DNA contains 6.6 into 10 to the power of minus 9 base pairs on whole. Okay, so students, from these values we can calculate the length of DNA. The length of DNA is equal to distance between nucleotides into number of nucleotides. Distance between nucleotides into number of nucleotides. Okay. So, the length of human DNA we are calculating. Human DNA length we are going to calculate now. We have studied the distance between nucleotides is 0.34 into 10 to the power of minus 9 meters into number of nucleotides 6.6 into 10 to the power of 9 base pairs. So, the length of human DNA is going to be 2.2 meters. Okay, the length of human DNA is 2.2 meters. So, students, here we observe that the length of DNA is 2.2 meters, and how this 2.2 meter DNA is going to fit into a cell which is having a size of 10 to the power of minus 6 meters. The size of a cell is 10 to the power of minus 6 meters, and the length of DNA is 2.2 meters. That means DNA is packed inside the cell. This DNA is packed inside the cell. Okay. In this chapter, in this concept, we will be learning about how this DNA is packed into the cell. Okay. First, let us see the DNA of Escherichia coli. Escherichia coli, E. coli. E. coli, it contains it is actually a prokaryote, right? It's a bacteria, it's a prokaryote. They don't have any distinct nucleus. Prokaryotes, they don't have any distinct nucleus. That means there is no nucleus is not present, so DNA is not present inside the nucleus. So, where is this DNA present? DNA is not even scattered inside the nucleus. DNA is present in the form of a structure which is called as nucleoid. DNA is present in the nucleoid. DNA is present in the nucleoid. Okay. So, this nucleoid actually consists of proteins. Okay. DNA is packed inside this nucleoid by using some positively charged proteins. Positively charged proteins. Okay, by using some positively charged proteins, DNA is packed into the nucleoid in E. coli. Okay, so students, here this DNA is in the form of large loops. DNA is packed in the form of large loops in the Escherichia coli. E. coli, DNA is packed in the form of large loops. Okay. So, this is about in E. coli. Let us see in case of human beings. Human beings show a very complex packing of DNA because their length is very long, right? Let us see how human DNA is packed into the cell. You know, students, there are a type of complex packing will be found, complex packaging complex type of packaging is found in the human DNA. Complex, very complex type of packaging is found in case of human DNA. Okay, very complex type of packaging is found in case of human DNA. This human DNA is packed with the help of positively charged proteins. Positively charged proteins called as histones. Okay, proteins, positively charged proteins called as histones are involved in the packaging of DNA. Okay, so these proteins are said to be positively charged because of the abundance of positively charged amino acids, because these histone proteins are having an abundance of basic amino acids. Basic amino acids. Basic amino acids are actually positively charged amino acids. Positively charged amino acids. Basic amino acids are actually positively charged amino acids. Okay. So, these amino acids are said to be positively charged, okay, because they have the free ends are having positive charges. Free ends of these amino acids, they have positive charges. Okay, histones are rich in positive amino acids like positive or basic amino acids like lysine and arginine. Okay, what is the histone proteins rich in? Histone proteins are rich in amino acids lysine and arginine. Histone proteins are rich in amino acids lysine and arginine. Okay, because of the presence of positively charged amino acids, these histone molecules are positively charged molecules. Okay, these histone proteins are positively charged proteins. Okay, so these histone proteins, they are occurring in the form of complex molecules called histone octamers. 8 molecules of histone proteins, 8 histone proteins together form histone octamer. Okay, so the DNA will be wrapped around this histone octamer. Okay, DNA is wrapped around this histone octamer. DNA is wrapped around the histone octamer. Okay, DNA is wrapped around the histone octamer. DNA is wrapped around this histone octamer, and when the DNA is wrapped around this histone octamer, it is called as nucleosome. It is called as nucleosome. This nucleosome consists of nearly 200 base pairs. Each nucleosome consists of nearly 200 base pairs. Okay, so 200 base pairs of DNA will wrap around the histone octamer. They form a nucleosome. In that way, along the entire length of the DNA, these nucleosomes are present. So, a series of nucleosomes forms a chromatid. A series of nucleosomes forms chromatin. A series of nucleosomes forms chromatin. Okay, so this nucleosomes in the chromatin will appear like beads on a string. The nucleosomes on chromatin will appear like beads on the string. They will appear like beads on the string. Okay, so this, in that way, in the form of chromatin, in the form of nucleosomes, a series of nucleosomes called chromatin, the DNA is packed. Okay, but during cell divisions, this DNA is further condensed. This DNA is further condensed to form chromatin fibers. Chromatin is further condensed to form chromatin fibers. This is generally occurring during metaphase of cell division. Okay, further condensation of the DNA from chromatin into chromatin fibers occurs during cell division, generally during the metaphase of the cell divisions. Okay, so students, at that time histone proteins are already involved in first packaging. So here, in condensation of the chromosome during cell division, in condensation of the chromosomes of the DNA, in condensation of the DNA during cell divisions involves another type of protein, called as non-histone non-histone chromosomal proteins, non-histone chromosomal proteins, okay, NHC proteins in short form, NHC proteins in short form, non-histone chromosomal proteins, non-histone chromosomal proteins are formed during cell divisions, okay. So, this is about few points related to the packaging of DNA in case of human beings, okay, in case of eukaryotes. In case of human beings, positively charged proteins, histones are present involved in a packaging of DNA. So, abundance of basic amino acids are there in these histones, that is why they are positively charged. Basic amino acids are actually positively charged amino acids, because positively charged amino acids means they have positive charges on their free ends, okay. And the rich on the one end of the amino acid there will be positive charge. Because these histones are rich in positive amino acids like lysine and arginine, these histone proteins are also positively charged. Now, these 8 histone proteins together form a histone octamer. This negatively charged DNA molecule will be wrapped around the positive charged histone octamer. Negatively charged DNA will be wrapping around the positively charged histone octamer to form a nucleosome. Each nucleosome consists of nearly 200 base pairs. Each nucleosome consists of nearly 200 base pairs. A series of nucleosomes form a chromatin. Chromatin will be appearing like a beads on a string, with the area of the nucleosome where the DNA is wrapping around the histone proteins appears like a bead, and the next place where the nucleosomes are again formed is appearing like a bead. So, in that way this is all appearing like a beads on a string, okay. Chromatin is further condensed during metaphase of cell divisions to form chromatin fibers. During that condensation of DNA during cell divisions, non-histone chromosomal proteins like NHC proteins are involved, okay. So, let us see about the condensation of DNA now, okay. So, students, you know DNA consists of 2 regions based on condensation, DNA consists of 2 regions, okay. Euchromatin, okay, which is a slightly less condensed region, it appears less dark during staining. It appears less dark in staining, okay. Whereas the other type of chromatin is heterochromatin. It is very highly condensed region of DNA. It is very highly condensed region of DNA, okay. It is very highly condensed region of DNA. It appears very dark in staining. Okay. So, students, euchromatin is the less condensed region, less dark region, which is appearing less dark in staining. Heterochromatin is highly condensed region, which is very dark in staining, okay. There are differences in function also. Euchromatin region, it is transcriptionally active. Transcriptionally active means from this euchromatin mRNA can be produced. Okay. Whereas heterochromatin is transcriptionally inactive. That means it cannot involve in the production of mRNA, okay. So, this is about packaging of DNA, okay. Then the next concept that we will be learning is the search for genetic material. Okay. Search for genetic material, okay. See students, during Mendel's experiments, okay, during Mendel's time, and also when the when this nuclein was identified, these two were done at the same time, okay. Proposition of Mendel's laws of inheritance, Mendel's laws of inheritance, and the presence of nucleoid, all of these have occurred at the same time, all these have occurred at the same time, okay. So, they were saying that there is some genetic material which is present in the people, which is present in the organisms, which is responsible for the inheritance of characters, and here the nucleoid was also identified that it might be the genetic material, okay. So, students, but the molecular basis of what is this genetic material, studies on this was started in 1926 only. Studies on molecular studies on this genetic material, molecular studies on this genetic material have started during 1926, okay, during 1926, okay. So, the observations made by Mendel, Sutton and Morgan, they have helped us to understand that chromosomes are the genetic material. They have helped us to understand that chromosomes are the genetic material. Chromosomes are the genetic material, okay. But students, the question is, what is this genetic material made up of, okay. The major question was, what is this genetic material, what is this genetic material made up of, okay, what is this genetic material made up of, this was a big question, okay. To solve this, first step towards this understanding what is genetic material was done by transforming principle, okay. We can understand how what is the genetic material was identified in steps, not at once by one person. So, it was observed by different persons in different ways. Let us learn more about it. Transforming principle. You know, in 1928, Frederick Griffith was working with a bacteria called Streptococcus pneumoniae, okay. During 1928, Frederick Griffith was working with a bacteria called Streptococcus pneumoniae. This is a bacteria which causes a disease called as pneumonia, okay. He was growing the Streptococcus pneumoniae, this bacteria, on a culture plate. He was growing them on a culture plate, okay. Then he observed two varieties of Streptococcus pneumoniae. One type which was showing a smooth, shiny appearance, and the second type was having a rough surface, one having a smooth shiny surface and one having a rough surface. So, he called this type of bacteria with smooth shiny appearance as S strain, okay. He called this bacteria with a rough surface as R strain, okay. See, this S strain, it was having the smooth shiny appearance because of the mucus, which is actually a polysaccharide layer, a polysaccharide layer on the surface of its body. So, because of that, S strain was appearing very smooth and shiny, okay. And he also observed that S strain is virulent. Virulent means it is disease causing. S strain is virulent, it is disease causing. R strain is not causing any disease. It is not causing any disease. Okay. he observed that R strain is not causing any disease, only S strain is causing disease. Okay. So, let us learn in detail what he did exactly to know what is a genetic material. Okay. So, in 1928, Frederick Griffith, he was working with a bacteria called Streptococcus pneumoniae, which is causing pneumonia. Okay. He was growing bacteria on a culture plate. He observed two types of strains. First type with a smooth shiny surface, it was called as strain. Second type with a rough surface, it was called as R strain. Okay. The S strain is having its shiny appearance because of mucus or polysaccharide layer on its surface. Okay. And he observed that S strain is virulent, disease causing, and R strain is not causing any disease. Okay. Then what were the observations made? What was this transforming principle exactly? Let us learn now. Okay. So, students, he observed a miraculous transformation. He observed a miraculous transformation. He observed a physical change in the bacteria. He observed a physical change in the bacteria. Okay. He observed a physical change in the bacteria. Okay. What was it? Let us see. So, what he did was he was performing experiments with mice. Okay. He was performing his experiments using mice. So, first he has taken S strain and he injected it to the mice. He injected it to a mice. Then the mice died. Okay. Then the mice died. Then he took an R strain. Then he injected it to a mice. It didn't die. It was alive. Okay. It was alive. Okay. Then when S strain was injected to a mice, it died, and R strain was injected to a mice, it was alive. So, he understood that S strain is virulent. It is disease causing. It is disease causing. Okay. R strain is harmless. It is not causing any disease. Not causing any disease. R strain is harmless. It is not causing any disease. Okay. So, now the next step what he did was he has taken killed S strain. He injected it to a mice. He injected it to a mice. The mice was alive. Okay. He took S strain and he injected it to a mice. This mice was alive. Okay. Then he took this killed S strain and alive R strain, okay, he mixed both of them, killed S strain and alive R strain, both of them together were injected to the mice. They were injected to the mice, both of them together, killed S strain and alive R strain. Okay. Then he injected it to the mice, and this mice, it died. Okay. When only killed S strain was given to the mice, it was alive. When killed S strain was given along with the R strain, this mice, it died. So, here he observed that there is something which has transformed, and one more thing also here was, in this dead mice he has isolated alive S strain bacteria. He isolated alive S strain bacteria. Okay. He injected killed S strain and alive S strain together into the mice, and this mice died. From this dead mice he could isolate alive S strain bacteria. He isolated alive S strain bacteria. So, from this he said that there is something which has transformed an R strain into an S strain. Okay. So, something has happened during this step which transformed an R strain into an S strain. Okay. So, this live R strains have become S strains. So he is saying that there is some transforming principle, which has been transferred from S strain into R strain, That is why this R strain has become the S strain. Okay, and he is saying that this transforming substance which has been passed from S strain to R strain is the genetic material. He is saying that whatever is passed from R strain into S strain into R strain is actually the genetic material. Okay. So, this was according to the transforming principle, which was explained by Frederick Griffith. Okay. Frederick Griffith has observed when he was making experiments with Streptococcus pneumoniae, which is a bacteria causing a disease called pneumonia. He observed a transformation, miraculous transformation in the physical condition of the bacteria. He observed that the S strain, when it was injected to a mice, this mice was dead. When R strain was injected to a mice, this R strain was alive. Okay. When because from this he understood that S strain is virulent, S strain is disease causing, and R strain is harmless. R strain is not causing any disease. That was observed by him. So, he performed further experiments by injecting killed S strain to the injected to the mice. Killed S strain is injected to the mice. He observed that this mice was alive normally, absolutely normal. Then he did what is, he took a mixture of killed S strain and alive R strain, he mixed them together and injected into the mice. And now this mice, in spite of having killed S strain also, this mice died, and from this mice he could isolate alive S strain bacteria. So, he observed that because anyhow killed S strain is there, so he observed that R strain has transformed into S strain. So, he said that there is some transforming principle which is transferred from S strain into R strain. This is the genetic material, okay, which is transferred from S strain into R strain. Okay. That was explained by transforming principle, but he could not say what exactly this genetic material is, whether if it is an RNA, DNA, protein or whatever it is, he could not explain what is this genetic material. Okay. He said that there is genetic material passing from one strain to another strain, but he could not say what this genetic material is exactly. Okay. So, now students, next thing is biochemical characterization of transforming principle. Okay, biochemical characterization of transforming principle. There were 3 persons who were called as Oswald Avery, Colin MacLeod, McCarty. So, these 3 people, they have tried to explain the biochemical basis of transforming principle. They have tried to explain the biochemical basis of transforming principle. Okay. So, the transforming principle was just explaining that there is something passed from S strain into R strain, right, and that is the genetic material. So, these people, they have tried to identify what this is exactly. Okay. So, they were saying that they have actually isolated DNA, RNA and proteins from the, they have actually isolated DNA, RNA and proteins from the bacteria, okay, and they have tried to make some experiments with it. So, what they did was, they used proteases, which are protein digesting enzymes, okay. They used proteases, the protein digesting enzymes on S strain, okay, on S strain. They used proteases, protein digesting enzymes on S strain. So, proteins of S strain were digested, but still this S strain was causing transformation, but still this strain was causing transformation, but this type of strain was still causing transformation. Then he used RNases. They used RNases on this strain. They are RNA digesting enzymes. RNA digesting enzymes on the S strain. They used RNA digesting enzymes again on the S strain. So then again still S strain could cause transformation. Again S strain could cause transformation. Okay, that means this genetic material is not a protein or it's not a RNA. Okay, then they have started to use DNases. DNases are actually DNA digesting enzymes. DNA digesting enzymes. DNases are DNA digesting enzymes. Okay, so students, when DNases were used on the S strain, they were used on the S strain, then it couldn't cause transformation. It just could not cause transformation. It just could not cause the transformation. Transformation didn't occur when the DNases were used. So from these experiments, these people have told that DNA is the genetic material. From these people, from these experiment, these people, they have concluded that DNA is the genetic material. From this experiment, these people have confirmed the DNA to be the genetic material. But these experiments were not accepted. This saying of these three people, Oswald Avery, Colin MacLeod and McCarthy, were not accepted by many of the people. Okay. Then came another people who have explained very clearly by performing some experiments. They have explained very clearly that DNA is a genetic material. Okay. So we have to learn also about how the genetic material to be DNA is being proved. Okay. So the genetic material is DNA. The genetic material is DNA was very clearly proved by the experiments done by two people. Okay, they are Alfred Hershey and Martha Chase Okay, Alfred Hershey and Martha Chase. These two people have performed some experiments which have exactly proved that DNA is a genetic material, and they performed these experiments during 1952. Okay, these experiments were performed during 1952. They have actually taken viruses infecting bacteria, which are called as bacteriophages. Okay, and they have done what is, they have grown these viruses. They have grown these viruses in two different mediums. Okay, one medium which was having radioactive phosphorus. So DNA contains phosphorus, no? So DNA contains phosphorus. So here in this medium, whatever viruses are there, they show DNA to be having radioactive material. DNA is having this radioactive material. DNA is radioactive. In the second medium, they used radioactive sulfur. Okay, DNA, it is not having any sulfur. No sulfur is found in DNA. So this radioactive sulfur is taken up by proteins. Okay, so in this case, proteins are radioactive. Okay, so in that way they have prepared two different types of viruses. One type of viruses in which DNA is radioactive, one type of viruses in which proteins are radioactive. Okay, let us see what the experiments they have done. Okay, So they have taken a bacteria to which they have okay, they have added this is for suppose if this is having radioactive DNA. Okay, so these viruses with radioactive DNA have sent this DNA, genetic material DNA is sent from these viruses into the bacteria. Okay, viruses infecting bacteria, bacteriophages, they will be generally acting in this way only. Bacteriophages infect the bacteria, they will send the genetic material into the bacteria, and this bacteria will be thinking that the genetic material coming from this this bacteriophage is actually its own type of DNA, and it will try to produce its own proteins. Okay, so actually what is happening with these bacteriophages is, before we learn this, we need to know what is happening with these bacteriophages. Okay, so these bacteriophages, they infect the bacteria. Okay, the bacteriophages, they infect the bacteria, they transfer their genetic material into bacteria, they transfer their genetic material into the bacteria, and bacteria will produce its proteins. Bacteria will be producing viral proteins. It does not know that it is a viral bacteria. So, viral genetic material, bacteria will be producing the viral proteins now. Okay. So, based on this experiment, they have taken bacteriophages as example. So, some bacteria were mixed with these bacteriophages. Bacteria were mixed with these bacteriophages. Okay. So, after they have allowed them to get infected, they have allowed the bacteria to be infected. They have allowed the bacteria to be infected.